In this video, we are going to go over hydrolysis reactions. Hydrolysis reactions are the most common reaction type in biochemistry. They are the reverse of acyl group transfer reactions. Water reacts with esters, thioesters, and amides to form carboxylic acids and either an alcohol, a thiol, or an amine. Water is the reactant that breaks the carbon heteroatom bond in these reactions. Hydro is water, lysis means to break. Hydrolysis are a major class of enzyme that catalyzes these biochemical hydrolysis reactions. So these are the three main hydrolysis reactions. We have ester going to a carboxylic acid and alcohol. We break this bond right here, and we add, well, we break the bond between the carbonyl carbon and the OR group. We add OH to the carbonyl carbon and a hydrogen to the oxygen of the OR group. We create a carboxylic acid and an alcohol. Thioester, we break the bond between the carbonyl carbon and the sulfur, and we create a carboxylic acid and a thiol. For an amide, we break the bond between the carbonyl carbon and the nitrogen, but we make, it goes one step further and we make a carboxylate ion and the conjugate acid of the amine. So the hydrolysis of carboxylic acid derivatives produces two products. The hydrolysis of an ester produces a carboxylic acid and an alcohol. A thioester produces a carboxylic acid and a thiol. An amide produces a carboxylate ion and the conjugate acid of an amine. So initially, the initial products of the hydrolysis of an amide is a carboxylic acid and an amine product. But those two products undergo subsequent neutralization reactions because amines can act as bases and carboxylic acid is an acid in which the carboxylic acid donates a proton to the amine, forming a carboxylate ion and the conjugate acid of the amine. So to predict the products of the hydrolysis of carboxylic acid derivatives, first you need to break the bond between the carbon of the carbonyl group and the heteroatom, the ester, thioester, or amide. So this bond right here, to produce two partial structures, an acyl group fragment and a heteroatom containing fragment. So your heteroatom containing fragment are the ones in purple or pink or whatever you want to call it, and then your acyl fragment are the same in the box. Then you turn the acyl group fragment into a carboxylic acid by adding a hydroxyl group from water to the carbonyl carbon, and you make a new carbon-oxygen single bond. Then you add a hydrogen atom from water to the heteroatom containing fragment to produce an alcohol, a thiol, or an amine. So adding a hydrogen to an oxygen, you make an alcohol, a hydrogen to a sulfur, you make a thiol, a hydrogen to a nitrogen, you make an amine. You're either going to make a primary or a secondary amine. In the case of an amide, after you've done that step, there is a transfer of a hydrogen from the carboxylic acid to the amine. So this hydrogen, the nitrogen takes the hydrogen from the carboxylic acid, and the carboxylic acid donates it to the amine, and you get the negative charge of the carboxylate ion. So this is the carboxylate ion, which is the conjugate, and then you have the addition of the hydrogen to the nitrogen making a positive charge on the nitrogen and a new nitrogen-hydrogen bond, and you've created the conjugate. So for practice, go ahead and pause the video, and then when you're ready, come back and go over it. So in this practice, we're going to draw the structures of the products of the reactions below. So the first thing you're going to do is you're going to break the bond between the carbonyl carbon and the heteroatom. The carbonyl carbon is going to become the carboxylic acid. So I'm going to draw that out. And then you add a hydrogen to the heteroatom to make an alcohol. So now we have the alcohol and the carboxylic acid. In the second one, we break the bond between the carbonyl carbon and the heteroatom. In this case, the aromatic is going to be with the alcohol, and it makes a phenol. And then the carboxylic acid is on the other side. With the methyl group attached on the other end. So we have the methyl, the carbonyl group with the alcohol, and then, or the carboxylic acid with a methyl group attached with a phenol. So application of hydrolysis reactions. Hydrolysis reactions occur in the cell and in industrial processes. The digestion of proteins and fats is an application of hydrolysis reactions. Acid-catalyzed hydrolysis of proteins with hydrolase enzymes, such as pepsin. Base-catalyzed hydrolysis of fats in the small intestines. And base hydrolysis of fats in soap making, which is called saponification. So the formation of soap or saponification, soap is the conjugate base of a fatty acid, and saponification is the hydrolysis of an ester in aqueous base. So you have triacylglycerol, where you have the ester groups. You have a strong base, and those break the bonds, and you're left with glycerol and three fatty acids connected to the glycerol. Vegetable oils are triacylglycerols, which upon heating in the presence of a strong base produce glycerol and fatty acid carboxylate salts. So notice that you have the carboxylate salt here. When you remove the glycerol from the mixture, you yield the fatty acid carboxylate salt only, and that is what's known as the soap. So the soap is the carboxylate salt, not the glycerol plus the carboxylate salt. So digestion of fats occurs in small intestines in mildly basic environments. Lipase enzymes hydrolyze fats to fatty acids, and fatty acid salts produce products transported to either, so the fatty acid salt products are then transported to either muscle or fat cells depending on the body's energy requirements. So they'll go to muscles when fatty acids are metabolized into carbon dioxide and water. In fat, the fatty acids and glycerols undergo reesterification reactions to reform triacylglycerols for long-term storage for energy. So for practice, we're going to write the structure of the products formed in the following saponification reaction. So go ahead and pause the video, and when we're ready, we'll go through it together. So in the saponification reaction, we break this bond and we create a carboxylate ion and an alcohol. So I'm going to just redraw this R group. We have our carboxylate salt. Plus our alcohol group. Let me move that over a bit. So we have our 2 carbons off the oxygen group. We have 2 carbons, oxygen. This is our alcohol, and this is our carboxylate salt. So in summary, carboxylate acid derivatives undergo hydrolysis reactions in water in the presence of a catalyst to form a carboxylic acid or carboxylate ion, and either an alcohol thiol or the conjugate acid of an amine. A saponification reaction is the base catalyzed hydrolysis of an ester, which produces a carboxylate ion and an alcohol. Saponification of a triacylglyceride or a triglyceride fat produces glycerol and 3 fatty acid carboxylate salts, known as soap. Lipase enzymes hydrolyze fats to fatty acids, and the fatty acid salt products are then transported to either muscle or fat cells, depending on the body's need.